So we are delighted to have with us today Professor Galia Avidan from Ben-Gurion University. Galia did her BSc in Life Sciences at the Tel Aviv University and then an MSc in Neuroscience and a PhD in Cognitive Neuroscience at the Hebrew University of Jerusalem under the supervision of Udi Zohari, while also collaborating with Rafi Malach from the Weizmann Institute. Afterwards, she went to do a postdoc at CMU with Marlene Berman, investigating developmental and congenital face recognition impairments. And afterwards, she returned to Israel and did another postdoc at the Tel Aviv Sourasky Medical Center jointly with Tel Aviv University, looking into clinical applications of fMRI. Following these, she then joined Ben-Gurion University, where she is now staff, a professor of psychology. She received multiple awards, honors, and scholarships throughout her career. She published dozens of papers, including in top leading journals as Nature Communications, Neuron, Annual Reviews of Visual Science, Current Biology, Trends in Cognitive Science, and others. Throughout most of her scientific career, she investigated high-level visual perception, including examinations of developmental, congenital, and acquired perceptual deficits, mostly but not only concerning face perception and visual expertise. Her research focuses on the psychological and neural properties of perceptual processes using a variety of methods, including behavioral, physiological, neuroimaging as fMRI, connectivity and structural methods, as well as computational approaches. In the last couple of years, she started to examine the internal physiological and visceral processes that are generally referred to as interoceptive perception, looking into subjective factors such as prior experience, personality, physical, emotional, and mental states. Today, we will hear about her research focusing on normal and atypical face perception. So welcome, Galia, to our seminar. We're delighted to have you with us.
Thank you very much, Alon, for this nice introduction. It's a little embarrassing. It's nice to be here. I've been participating in a number of these seminars, which are great. So thanks for organizing this great forum. And nice to see familiar faces and also new faces. Hi, Rafi. And today I'm going to present work that kind of sums up a lot of research that was conducted, started long ago in my postdoc together with Marlene Berman. And over the years, we continued to work on the topic of congenital prosopagnosia. And the work that I'm going to present today is kind of a sort of a review of our work and additional work from the literature that we kind of comprehended, and we try to suggest a new sort of account, a new theoretical account to think about congenital prosopagnosia based on this sort of long line of research over the years. So I guess most of you are familiar with what congenital prosopagnosia is, but just in case it's new for some people in the audience, I'd like to share this video clip. This is an interview with the painter Chuck Close, who recently died. And he apparently suffered from congenital prosopagnosia, so face blindness. Meaning that he had great difficulties recognizing people, even of very, very familiar individuals. And it's sort of quite interesting to hear him, particularly given the fact that he mainly focused on painting portraits. So it's kind of an interesting sort of choice of a career for someone who suffers from prosopagnosia. So let me just play this short video from an interview with him, and then we'll continue.
In the face blindness, the name is how do you say it?
Prosopagnosia.
Yeah. Now that means that you can't recognize faces, you have trouble?
It's a sliding scale.
Yeah.
I have a great deal of difficulty recognizing faces, especially if I haven't. If I've just met somebody, it's hopeless. I will never remember them again. unless it's reinforced over and over and over. And even people that I know very well, if I haven't seen them for a while, it's like a bucket with a hole in it. Information is coming in, but it's pouring out the bottom just as fast. And I'm often losing information.
But you are known for portraits of faces.
I was driven to make them. I'm absolutely positive.
That's what did it.
Because in real life, if you move your head a half an inch, to me it's a whole new face I've never seen before. But if we flatten it out, I have, and I take photographs, I work from photographs, and I make flat things called paintings and prints. I have virtual photographic memory for anything that's flat. So it's not an accident that I only do images of people who matter to me, family, friends, other artists. There are no commissioned portraits.
Okay, so there are some very interesting sort of issues that come up in this interview, how it's really kind of a sliding scale, so it's not an all or none phenomena, but then there needs to be many iterations of a face in order for these individuals to be able to recognize them, and they may even fail to recognize very close relatives. And so in a number of studies that we conducted, as well as others, we've seen that individuals with congenital prosopagnosia actually exhibit a sort of a similar behavioral profile to individuals with acquired prosopagnosia, and I will later on remind a few examples. However, importantly, in contrast to acquired prosopagnosia, these individuals do not have any specific cortical lesion that can account for the deficit. As far as we can tell, it's present from birth, despite the normal opportunity that these individuals have to acquire face processing skills. And we see reports about the deficits running in families, implying that it has some genetic underpinnings. The genes are not yet identified, but there are some studies that looked at it. There's a recent study talking about a specific gene associated with calcium binding membrane protein that is specifically expressed in the central nervous system, but we still don't know the exact genetic underlying mechanism here. And there are also arguments in the literature regarding the exact prevalence, which is sort of a long discussion. Right now, the numbers kind of ranges between 1% to 2% of the population. And a very important aspect of congenital prosopagnosia, which is always important for me to stress, is that this is a deficit that is evident in real life. So even though in real life, these individuals interact with other people and hear their voices and see their body and have a chance to extract any relevant information from a person, they still fail to recognize these individuals, kind of implying the priority that the visual information from the face has for person identification. And so what I want to do today, as I alluded to at the beginning, I want to start by providing a brief overview of the literature regarding the behavioral and neural characteristics of this disorder. And then I will try to integrate these findings with some additional perspectives coming from physiology and computational principles, and then kind of tie it all together to this new framework that we would like to suggest, which also has obviously lead us to predictions and future directions. So I'm hoping to kind of take you through this logic, and as Sharon mentioned, these ideas were recently published in a paper Marlene and I wrote for the annual review of Vision Sciences. So the behavioral findings, first of all. So the main impairment, of course, that we see in congenital prosopagnosia is with recognition of famous faces. And this is evident in different tasks where they are shown pictures of faces and have to identify to respond, to provide the name or other contextual information. But again, we also see evidence for impairments not only in tasks requiring recognizing familiar faces, but also just perception and short-term memory of unfamiliar faces in various tasks. And the kind of most well-known task that is shown here is the Cambridge face memory task that was devised by Brad Duchesne and is widely used in the literature also for diagnostic purposes. But again, it's not only memory, because even when faces are just presented on the screen simultaneously, these individuals show deficits. And of course, an immediate question that should be asked is to what extent these deficits are indeed specific only for faces. And in a recent review paper that was done in Marlene's lab, they gathered data from many publications in which some information was available regarding object recognition in congenital prosopagnosia. And we can see the overall results here. Basically, the main finding is that in at least 66% of the individuals for whom there was enough data to analyze this question, there appears to be also a deficit in object recognition. So it's kind of, again, it's a mixed phenomena, presumably, with 66% of the individuals tested showing some deficits in object recognition as well. But then there's this other third of individuals who show clear deficit only in the domain of faces. And these are the current findings. And of course, this requires further investigation. And the difficulties in face processing are typically attributed to deficits in holistic perception. And this was shown, again, by a number of groups, including our data, using different paradigms. Here, there are some examples: the upright inverted paradigm, the composite paradigm, and a paradigm in which there are spatial variations within the features comprising the face. And in all these variations, there's evidence showing impaired holistic processing for faces in congenital prosopagnosia. But even kind of more intriguingly, there are also studies showing that these individuals show impairments in other tasks associated with configural or holistic processing, even when faces are not involved. So again, going to this notion that there might be a more general impairment in holistic processing as long as the processing is required for face perception. And obviously, because holistic processing is sort of disproportionately required for face processing compared to other objects, we see a stronger deficit in face perception compared to the perception of other stimuli. So that's in the behavioral domain. And again, I'm kind of giving a very broad description. And there are numerous studies that I did not include here, but I'm just kind of trying to lay out the basic concepts for to follow on in the rest of the talk. So what about
Galia,
the neural?
Can I ask a quick question?
Yeah, hi, Galia.
Because you showed, hi Galia. So you showed the Chuck Kloss interview, and his paintings are actually mainly based on like holistic processing. I mean, unless you would define it differently, because you know, he's painting all those different parts in different colors, and then when you look at it from a distance, you see a face, right? So I mean, how would you consider this type of presentation?
So yeah, so his paintings are special in the sense, as you say, that it's actually, they are comprised of multiple very small elements, like little features that only when you kind of add them up become sort of a face. Mm-hmm. But I take it actually as evidence for his kind of picture-based, feature-based processing, because what he did is basically just to copy or to take elements from the photographs that he had, and then just sort of comprising multiple elements kind of in a feature by feature fashion, rather than just, you know, drawing like a global face and then filling in the details as maybe other painters would do. But this is sort of my interpretation.
Yeah, right. You can think about it the other way around. And you also said something about like a 2D versus 3D, right? Perception, that in 2D it's easy for him, and the 3D is where it's difficult. But, you know, prosopagnosia is tested on 2D images, right? And we typically see it in both cases, right? So this is not also not.
Yeah. So in lab settings, of course, we mostly studied 2D images. In real life, it's 3D, and they show individuals show deficits in both. This is sort of his own experience. I have to say he was never kind of formally diagnosed. Okay, so it's kind of a self-testimony, which is interesting, but I don't think we don't know for him whether it was true that it's 3D or 2D.
But there's no study that looked at that.
About the 3D, he was talking about a time change, that when the image changes, the rotation, then the new image is different and is not recognizable. So
this is another thing.
To the image, he was freezing it.
Yeah. So it's kind of thought-provoking to hear his own insights, but again, it was never kind of validated on him in a more rigorous way. So it's but I agree. It's very interesting, and this is why I chose to show this clip. So just to continue to the neural findings, and again in kind of very broad brush strokes, and just to sort of remind, we have the basically when we look at normal face perception, we know that we can roughly talk about a core face system and an extended system, and this is kind of an older perspective on how we devise the core and the extended, and there's some more recent work, some of which by Galit and others, suggesting that we can think about the core and the extended system in a somewhat different way than the one depicted here. But again, for our purposes, we started by looking at whether we could find any sort of alterations in the core system, again, coming from the perspective of acquired prosopagnosia, where the deficits are located in these core regions for the most part. And again, in a number of studies along the years, we've shown multiple times that there are actually normal activation in congenital prosopagnosia in the core regions that were normal in terms of the anatomical location, the selectivity to faces, and the size of the activated foci. And from a different perspective, or sort of from a complementary perspective, we found evidence that there's actually impairments not in the core system itself, but actually in the connectivity of regions in the core regions to more anterior part of the temporal cortex and the inferior frontal cortex. And this was first shown in a study where we used diffusion tensor imaging. And specifically, we focused on the inferior longitudinal fasciculus that starts from the occipital cortex through and connects it to the regions there to the anterior temporal cortex, and the inferior frontal occipital fasciculus that connects all the way to the frontal cortex. So we know that these two white matter tracts are important as they kind of traverse all the sort of ventral stream, basically. And we see here that in this analysis, the y-axis show the fractional anisotropy in these fibers. And here we see the fibers on the right hemisphere and the left hemisphere. And we see that for both hemispheres, we see here kind of a global deficit or global difference between the structure of these tracts in individuals with congenital prosopagnosia in gray compared to the controls. And there's also a very modest correlation here between the performance of these of the participants and the mean fractional anisotropy. And the results was similar results, or results along similar lines, I should say, were also found by others. This is another study, not from our group. And here they suggested that the deficit is also in the white matter fibers is more related to local white matter that is adjacent to regions in the fusiform gyrus. So we can see, and mostly in the right hemisphere. This is what is shown here in this graph. And again, the results showed modest correlation such that the better the sort of diffusion measures, the better was the facial recognition skills of the participants. And in a more recent study, this line of results were also shown by the group of Congenital Spatial. And in their study, they found deficits in both the sort of whole global structure of fibers connecting the middle fusiform area, but also in and more pronounced evidence for local white matter abnormalities in these regions. And again, here there was a correlation, but only in the typical participants, such that better performance was correlated with the local measures of the fractional anisotropy. This is on the Benton task and the CFMT task. So again, generally speaking, we see that there are alterations in the white matter connecting occipital areas to the area of the fusiform gyrus, and presumably also more anteriorly to the anterior part of the temporal pole, and even to the frontal cortex, as we saw in the inferior fronto-occipital fasciculus. In parallel, our studies also looked at aspects of functional connectivity, so not only structural. And from in these studies, our focus was not so much on one particular connection, but rather looking at the sort of general topology of the face system in typical individuals compared to individuals with congenital prosopagnosia. So kind of taking a zoom out perspective and not examining specific regions or specific connections. And I want to show you the results. Let me just stop this for 1 second so that I can explain what we're looking at. So in this analysis, what we've done is to compare a group of congenital prosopagnosic individuals to controls, to matched controls. And what we see, like the video is not obeying my, just trying to stop it, but it's not. So it will continue to flip. So basically what we did here is to look at all the connections which would be that are stronger in individuals, in normal individuals, compared to those connections that compared to individuals with congenital prosopagnosia, and to look at the opposite contrast. So what are the connections that are actually stronger in individuals with congenital prosopagnosia compared to control? So it's kind of what I'm showing you here is a contrast between these comparisons. And basically the red circles that we see here are nodes related to the face processing system, or area nodes that showed face selectivity. The green are nodes that showed general object selectivity, and blue nodes are those that are just kind of non-specific. And the red lines indicate the edges that connect the different regions. But in each of the comparisons, it's basically those edges that are stronger in one group compared to the edges that are stronger in the other group. So let me just start this video again, and you can see how it flips from the difference between controls and prosopagnosics to the difference between prosopagnosics and controls. And the notion here that in controls, we see the activity, the connectivity to the anterior temporal cortex, mostly in the right hemisphere. This is this big focus here, this node that actually exhibited sort of a hub-like profile in terms of the network organization, while in prosopagnosia, we see a very, very different picture. So what we see here is that the network is connected to many regions in the more posterior areas of the brain, and the nodes that are more dominant in this network are actually more object selective than face selective. And the node in the anterior temporal cortex is completely missing in this contrast, meaning that for congenital prosopagnosia, it's kind of not playing a critical role, or is not a critical part of the topology of the face network. So this to us was a very important result in the sense that it showed how actually congenital prosopagnosia is sort of changing or affecting the topology or the organization of the face network as a whole, on top of potential differences in specific regions that comprise this network. And of course, we don't know whether this result is due to potential compensatory mechanisms. So presumably, or it could be that the lack of this hub in the anterior temporal cortex result in this compensatory mechanism, which elicits this hyperconnectivity in the more posterior regions of the brain, or it could be maybe the cause. So maybe because they don't have this, this posterior hyperconnectivity leads to the poor connectivity to the anterior regions. Of course, we don't know the result. But what we did find is, again, an interesting correlation, such that the greater the hyperconnectivity was, the better was the recognition abilities for faces of the prosopagnosic subjects. So basically, those who showed better performance, everybody were impaired, but those that were better actually had greater hyperconnectivity in these posterior regions. So this alludes to the notion that it could be some compensatory mechanism. So they are actually processing the information about the faces using these more posterior regions. Of course, this is not efficient, so they are still prosopagnosic, but it brings them, or it enables them to process the faces a little better. So these are the results from our studies. And again, there are some other studies that have looked at this using a network approach, but using very different methodological tools. Here, this is another study which showed impaired connectivity using seed-based correlation analysis. Here they seeded in the right occipital face area and showed impaired connectivity to areas in the core system. And here it's an example where they seeded from the right fusiform face area and showed impaired connectivity to regions in occipitotemporal cortex, but also more anteriorly, as is shown here in this summary figure. And again, also in this work, there was an interesting correlation to behavior. The analysis was, again, very different from what we've done. And we see here the impaired performance of their group of individuals with prosopagnosia compared to controls on a long-term memory, face memory task. And they used the impaired connectivity in a model to kind of predict the level of face processing. And what is on here is the correlation between the predicted value of face processing based on the model to which they fed the impaired connectivity and the actual performance of the participants. So sort of a different perspective, but conveying a similar notion. So taking all these results together, we want to come up with sort of a working hypothesis regarding prosopagnosia. And basically what we suggest is the following. So the abnormal connectivity between the core and the extended region is related to and might underlie the abnormalities that we see or other abnormalities in behavioral, physiological, and computational characteristics associated with prosopagnosia, which I will explain in the next few slides. And there is an assumption here, and that is that we refer to the perturbed cortical topology that I described as the underlying deficit, and we assume that all the other deficits that I will show in the next slides are sort of a consequence of that. And again, this is a working hypothesis that I'm suggesting, and we don't have direct evidence to support it. We don't know whether this is the case or not. It's just sort of the hypothesis that we try to put forward. So what are those other deficits that I'm referring to? They're basically in a number of domains. So the first deficit that I would like to focus your attention on is related to receptive field properties in individuals with congenital prosopagnosia. So receptive field properties are sort of a very basic, prominent aspect of how visual information is being processed. And we know that receptive field properties change along the areas of the ventral pathway. And there's very interesting work from Kalanit Spector lab, who looked at the receptive field properties in individuals with congenital prosopagnosia. And this slide shows how the receptive field properties change in typical individuals compared to prosopagnosic individuals. The typicals are in blue and prosopagnosic in red. And the y-axis is the receptive field size, and the x-axis here is the eccentricity. So we want to see how the receptive field size changes along the entire eccentricity. And we see some interesting results here. So basically, in individuals with congenital prosopagnosia, and this will be kind of more clearly shown in the next slide, we see a deviation from the normal increase in receptive field size that we typically see in normal individuals. So basically, we know that receptive field size are very small in V1, and then they gradually increase in size in V2, in V3, until they reach a very large dimension in the anterior part of the sort of ventral pathway. But we see that in congenital prosopagnosia, this is not really the case. So the receptive field size show this deviation from the typical increase somewhere along here, and definitely in phase selective regions. And I want to show you this sort of from another perspective. This slide here, again from the work of Congenital Spector, shows the visual field coverage. So we see that in controls we have, and also in prosopagnosic, we have sort of similar visual field coverage that is obtained by those receptive fields in early areas of the ventral pathway, V1, V2, V3, and then starting sort of in around V4, we see a somewhat deviation from this typical sort of pattern that we see in controls. So in everybody, also in controls, we see that in phase selective regions, we have coverage that is associated mostly with foveal regions, but we see also good coverage of the rest of the visual field, which is again related to the size of the receptive field. But in congenital prosopagnosic individual, we see a very, very restricted coverage that is really tiny of the visual field. It's really only the fovea and not other regions of the visual field. So what we, or the way we think about it is that actually this difference in receptive field size and the sort of related visual field coverage is sort of a fundamental difference, which is related to the way that the spatial pooling of information is done across the ventral pathway hierarchy. So we know that information is being passed from one region to the other, and there is some sort of spatial pooling that is taking place and is related to the fact that receptive field size grow larger and larger. So each area is receiving information from adjacent spatial location that are covered by the preceding area. And we also know this, or we also think about this in relation to, for example, recent computational models such as deep convolutional neural networks, which try to suggest model for the ventral pathway based on such networks. And we know that also in these networks, the component, the spatial pooling is very critical. So again, kind of borrowing the concept of receptive fields also to these models, we basically think of neurons in upper layers in the model as those that sample larger portions of the visual field with more with some special kind of overlap between neighboring neurons, and this is again embedded in the normal connectivity in the ventral stream, but also in the deep neural network models. And such spatial overlap allows efficient sampling of the environment, and it also allows some generation of predictions. So if we're kind of located or focusing on one area of the visual space, and we sample it in an efficient way and transfer the information to the next area of the ventral hierarchy, it will allow us to generate some predictions as to what would be the next area on which we should focus. And again, this will become soon kind of evident, or the relevance of this would hopefully become evident in the next few slides.
Galia,
yeah.
on the previous slide, could you tell us what the scale is in degrees? The previous slide.
Previous slide. This one?
Yes. Yeah. What is the scale in degree?
The scale.
I realize this is in millimeters or whatever the units are on in the imaging, but this circle in V1, say, what does it cover in degrees? It's visual field, you're saying, so.
Yes, I don't have it like in the
Yeah, well, roughly, I mean, is it 10 degrees?
It's
100
yeah.
degrees?
I will
Yeah.
check. I will check while Galia is talking and get back to you.
Thank you.
It's in my notes, but I don't have them in front of me right now. So it's several degrees, but I don't have the exact number. So I don't want just to give you like a wrong answer on that.
That's fine. Go ahead.
Okay, thanks. So yeah. So in terms of, again, thinking about this notion of receptive field size and spatial integration, we need to, again, go back to behavior and see what these kind of physiological mechanisms, or what is the relevance of these physiological mechanisms in terms of behavior. And we want to try to link them to holistic processing, which is, again, so critical, as we already know, for normal face processing. So in terms of the studies that have looked in these parameters, we can think of the notion of receptive field size if we take it to behavior, and as sort of the flip side of that would be what would be the optimal distance from which we should look at a face in order to be able to process it in a holistic fashion. And relatedly, again, if we think about it from a physiological or from a computational perspective, we need to think about what would be the sort of optimal neural tuning size. So what is the optimal sort of parameter in for example, deep neural network model that would account for this increase in receptive field size and for this spatial pooling that I was talking about in the previous slides. So one study, or a couple of studies that looked at optimal distance size, there are not too many, which is quite surprising, but I want to show you quickly some results from the studies who did. This is an interesting, a nice sort of illustration. And I hope you can see there are two people here kind of approaching to each other, and the notion here that is from far away, the processing is not going to be related to holistic perception because the face is just not really kind of evident. But as they kind of come close together, there is this optimal distance, and the notion from the studies, mostly from this study from Eleanor McCone, is that there's the optimal distance that ranges between 2 and 10 meters in which holistic face processing takes part. And this is illustrated here. So there's this kind of inverted U-shape function showing that indices of holistic processing that they collected using different paradigms are actually at the peak here in the viewing distance of 2 to 10 meters, and they drop in shorter distances, but also in longer distances. So there's sort of this optimal range in which a face should be presented in order for us to be able to process it in a holistic way. And again, the same notion was also demonstrated in the computational model, where they used a parameter that they called the neural tuning parameter, which again is basically sort of this parameter determines the extent of the connectivity between the neurons in sort of intermediate level of the visual hierarchy and the areas in the highest level of the hierarchy. And what they showed is that this parameter reveals optimal processing of the model only when it's sufficiently large. So they used they tested their model with the composite task, with the upright inverted task, and with the part whole task, all kind of gold standards for measuring holistic perception. And their notion is that the performance of the model was optimal in sort of resolving these various tasks, accounting for holistic perception, when they used a large neural large parameters akin to large neural tuning, and again related to the extent of the spatial pooling. So spatial pooling, according to this model, is allowing, or sufficient spatial tuning is allowing more optimal holistic perception. So just to summarize where we are up to now, and I hope this was not too much, because there's still a little bit more in order to sort of fully understand where we're heading with these results. We saw that optimal face processing is related to spatial integration, and we saw impairment in spatial integration in terms of, we saw impairment in holistic processing in congenital prosopagnosia. I suggested that spatial integration is the outcome of the receptive field size in the normal brain, and in congenital prosopagnosia, we're suggesting that this spatial integration is actually impaired due to the abnormality of the receptive field size in areas related to face processing. And we also saw evidence for impaired connectivity. And again, we think, or we'd like to suggest that the deficit in receptive field size and the abnormal spatial integration are actually consistent with these deficits in connectivity in congenital prosopagnosia. And this brings me to another dimension of behavior, which is very important, and again related to eye movement, to spatial pooling or spatial sampling of the environment, and to receptive field size. And this is the eye movements. So eye movements are basically the way in which we sample the environment. So we're mostly not just static staring at a single point, but actually continuously sampling the environment, including faces. And so if we take into account everything that that we talked about so far, and think about what we expect from eye movements, or what would the patterns of eye movements would be like in congenital prosopagnosia, we can come up with the following predictions. So if indeed the spatial integration is reduced in congenital prosopagnosia, then we expect that each fixation would only provide sort of relatively local information, right? Because the receptive fields are small. So even if they fixate on the right location within a face, the information that would be available for them is going to be much more restricted. And so we expect that they would exhibit more fixation, and more fixation would actually be required for them in order to perform face perception tasks. But because the receptive fields are small, even though they will perform or conduct many more, would show more fixations, their overall integration would still not be sufficient to elicit normal perception of the face. So it's not that they can just collect a lot of information and just sort of by that compensate for the deficit, because wherever they look, it's going to be a very small sample of the visual environment. So even if they sample it with multiple fixation, it's not going to suffice. And again, if we relate all these parameters together, we expect that this deficit would be, again, a general deficit in holistic perception, which would be more pronounced for faces compared to other objects. And here again, I'd like to show some results that were conducted in other labs, not by us. And there are multiple studies that looked at eye movements and face perception in congenital prosopagnosia, and I cannot, of course, cover them all. So this is not an exhaustive sort of review, but this illustrates nicely the points that I wanted to allude. So what we see here is data from one such study by Malaspina, and they showed in a task where participants looked at upright and inverted faces, they show exactly the results that are predicted based on our kind of working hypothesis. So participants showed more fixations. The congenital prosopagnosic individuals showed more fixations, but the fixation were significantly shorter. Their saccadic length was also shorter. And there were also some interesting findings related to the fact that participants looked at upright and inverted faces. So again, alluding to the notion of holistic processing, if we just look at the sort of overall pattern of eye movements exhibited by these individuals, and the color here denotes where the maximal time was spent while looking at these images, we see that control participants in upright faces were mostly kind of focused in the eye region and somewhat on the nose. But for the inverted faces, the pattern was very different, with more fixations also in the region of the face, of the mouth, sorry. But for the congenital prosopagnosic individuals, there was no difference kind of overall when looking at upright versus inverted. So for them, also when examining upright faces, we see fixations on the face, on the mouth as well. Again, alluding to the abnormal way in which they process upright faces. And again, interestingly, a somewhat similar pattern was also evident when they examined flowers. So this was a very sort of demanding task using very ingenious stimuli like the flowers that are shown here. And again, we see more fixations, which were actually shorter. So kind of resembling the notion that we put forward, that the general sort of pattern might also be evident in other objects, not only on faces. And now I just want to kind of tie it all together eventually and present the general notion that we'd like to propose, this sort of new framework that we try to propose for understanding congenital prosopagnosia based on all this literature that was, all these findings that was gathered by us and others over the years. And just this sort of, we're trying to link face perception, which is based on areas in the ventral stream, to things like eye movements, as I alluded to in the last couple of slides. But how do we do that? So the ventral stream, we know, is kind of more associated with identity processing and is thought of as being more tolerant to stimulus-specific information, while eye movements are very specific. They involve action, they involve sort of active gathering of information from the environment, and they should be very precise, right? There are much more kind of analytically executed than the way we think about the ventral stream. So how do we tie these two things together? And for that, we're actually turning into sort of an interesting area that for a long time has been kind of out, or areas in the brain that were kind of outside the focus of face-related research. And these are the regions in the entorhinal cortex. So in the medial part of the temporal pole, the temporal lobe. We know that in rodents, there are those unique cells, the grid cells, that are found in the entorhinal cortex and in other regions as well. And what is special about these cells is that they actually encode location of, this is in rodents. So it's basically while rodents are kind of actively sampling the environment or kind of foraging the environment where they are. And we can see in black the traces of their foraging behavior. And in red, we see the spiking activity of neurons recording in different parts of the entorhinal cortex and also the dentate gyrus in the hippocampus. And this elicits these unique kind of hexagonic patterns which receive the name the grid cells. And how is this related to our story? So the interesting thing is that similar patterns of responses were also found in cells in the entorhinal cortex of non-human primates. So not only in rodents, as was initially found, but also in monkeys. But in monkeys, the critical difference is that this pattern was evident when the monkeys actually performed eye movements. So this is more typical to primates, right? We don't just kind of run around in our environment, not us and not also the monkeys, unlike the rodents. So our way of sampling the visual environment, or the most natural way, is with eye movements. So here we see recordings in the entorhinal cortex of the monkey. And we see traces of eye movements on different stimuli presented to the monkey. So the monkey is stable, okay? It's just the eyes that are moving. And interestingly, we see sort of typical hexagonic patterns as was found in the rodent brain during active locomotion. So the response in the entorhinal cortex in the monkeys are kind of similar in notion to the grid cells in rodents, but again, critically, they are obtained during tasks that involved eye movements and not locomotion. And the implications are that, again, the spatial representation of the visual stimuli could be encoded while visual exploration is done at a distance from the stimulus, which again, as I just said, it's more natural for monkeys and also definitely for humans, and is potentially associated with the way we explore faces. And importantly, there are also some fMRI studies that looked at grid-like patterns in the entorhinal cortex in humans. But we don't have time to go into these studies. And relatedly, there are, again, a recent computational model by Bikansky and Burgess actually proposed exactly this notion that these grid-like cells in the entorhinal cortex of primates and of non-human primates, but also potentially in the human brain, could actually account for high-level visual processing. So kind of an extension of the ventral stream that connects the notion of sampling the environment using eye movements. And this computational model basically suggests that the grid cells, or we should call them the grid-like cells, because we don't know whether they are exactly like the grid cells in the rodent brain. The grid cells basically encode translation vectors between salient stimulus features. So these is like one point of fixation, and then the next point of fixation, and the following point of fixation. And these would be salient features in the stimulus, like in the kind of classic recordings of eye movements when sampling a face. And what we see here in the model is basically sort of the alluding to the pattern that such encoding would elicit in entorhinal cortex. So a sequence of saccadic eye movements vector moving from one salient feature to the next would enable us to potentially confirm sort of a general hypothesis, an initial hypothesis that we have about the identity of the stimulus and its relative relative to the layout of the scanning. And this model is really sort of the first, this computational model was the first to suggest kind of an explicit mechanism for the view that saccades actually supports kind of hypothesis-driven construction of perception and recognition. And it's very relevant for face processing, but also for processing other visual information, of course. And it's compatible with holistic perception. And for us, it's sort of a very kind of important step in trying to understand how all these potentially or these different kind of parts of the puzzle that I alluded to are actually connected together. So in this last couple of slides, I just want to show the model that we suggest and how we tie all these different pieces together. So this is just a kind of a schematic representation of the occipital areas, occipitotemporal areas related to face processing, going to the more anterior areas, which include the anterior temporal lobe and the amygdala and potentially inferior frontal gyrus. And on the medial view, what we also want to stress is the entorhinal cortex that is located here in the medial part of the temporal lobe. And as I described in the previous slides, we think that this area is important for the spatial integration of information from the face. And this integration can be done using basically eye movements. And I'm also alluding here to this notion of the spatial integration along the ventral pathway, as is done in the biological system, the brain of monkeys and humans, but also, as I suggested, in models of deep convolutional neural network. And sort of at the end of the hierarchy, we see some representation which is based on kind of a multiple kind of a face space representation that is based on multiple dimensions. And again, this was suggested also based on behavior in the human studies, but also in recent findings from Doris Tsao, for example, in the monkey brain. And again, the impairment in congenital prosopagnosia, as I alluded to throughout the talk, is related to the fact that the spatial sampling of the space is impaired due to the small size of the receptive fields in areas related to face processing. So just to sum up, our proposed framework is consistent with evidence showing that the hippocampal entorhinal circuits are involved in organizing spatial, but also non-spatial information into some relational and matrix configurations. And here we suggest that faces are sort of another example such relational information. And such configurations capture low dimensional geometry. And in that sense, again, are also relevant to information about the face. And this framework implies that holistic perception is embedded in, and the impairment in holistic perception are actually embedded in neurobiologically and computationally tangible underpinnings. So it's not just talking about holistic perception in kind of abstract, in an abstract way, but we also try to see how they are actually implemented in those computational and neurobiological models. And of course, these ideas now needs to be tested more formally, and they elicit a number of different predictions and questions for future research, which we can discuss. But before we do so, I'd like to sort of thank everybody who is involved in this project. So first and foremost, Marlene Berman, with whom all this work was done, and with whom together we wrote this review paper. So great thanks to her and my students, current students, and past students in the lab that contributed to the work on congenital prosopagnosia, and Olaf Sporns from Indiana University, with whom I collaborate on issues of network properties. And finally, Ulrich Hasson and Ariel Goldstein from Princeton University, that spent many hours discussing these ideas with me when I was in a sabbatical in Princeton. And thanks to you and for listening. And I don't know if we still have time for questions or if we have time to talk about predictions. I'm happy to do whatever.
Thank you very much, Galia. I'd like to ask everybody to unmute yourselves and give you a big applause for a very intriguing and interesting and thorough talk. Very interesting. And yes, we have time for questions if you do.
Sure.
Okay. So the floor is, yeah, it's open for questions. You're most welcome to ask. And
can I ask?
Yeah, sure.
Okay, Galia, thanks. That was really fascinating.
Thanks.
So I always interpreted your previous finding about this connection between posterior and anterior areas, which made a lot of sense, given that the FFAs seem to be normal in prosopagnosia and everything. That it was a disconnection between perceptual and semantic processing. Mm-hmm. And actually, what you're saying now that it's all perceptual, right? It also the anterior, aspects of processing is, you explain it based on the eye movement and the sampling. So is that what you're thinking right now? We can explain everything based on the perceptual representation, and we can drop this semantic part.
It's a great question, and it's something that it's a question that I'm also, and me and Marlene, I think we're both also kind of struggling with this, because it's a bit extreme to some extent, right? But it's also interesting if we can really account for everything that we see, and we think about high-level processing and semantic processing as being rooted in very fundamental physiological properties of the visual system. So I don't know. Like the short answer is that I'm not sure, and I don't know whether we could sort of put all the weight only on that, on having some sort of impaired representation at the very basic visual level, and sampling of the environment and eye movements, and whether this could account for the entire deficit or not. But to me, it's kind of, it was interesting to think about whether we could actually come up with such model. So don't talk so much about the high level sort of in itself without trying to tie to the basic physiology. And I guess, Rafi, this is due to your training. But I don't know, and it's an open question, really. Yeah.
Yeah, I have to. Okay, so I want to. I think it actually all connects. Because I would want to suggest that there is a semantic representation, or you can say multisensory representation of faces, but maybe along with Galia and Marlene's model, there's a disconnection between the visual perceptual representation and the other semantic and other sensory modality representation. So the other representation exists, but the deficit may be in the fact that they're not tied together in some way. Okay, just again, hand waving, but.
Galia, can I say?
Yeah, sure.
Very fascinating. I never thought about this grid possibility in relation to face. Beautiful. But I have a question about the eye movement, because I'm curious. I mean, we obviously can recognize famous faces if they're very famous and very clear with a flash of, let's say, 200 milliseconds without the need to move our eyes. So are you suggesting that if I let you scan images, then I'm greatly improving in my ability to recognize them, to do invariant processing? I mean, is it known? I mean, is it
we have, yeah, so this is, it's a very important point. So and I didn't go into that, but basically what we're suggesting and other studies have shown is that eye movements are very important during face encoding or during learning of new faces, and are not as important when viewing faces that are highly familiar. So for a highly familiar face, you're right. I mean, you're done with like 1 or 2 fixations. You don't need more than that. And Yoni has some interesting work showing differences in eye movements for familiar versus unfamiliar faces. So, but it's true. I mean, for faces that are highly familiar, you don't really need eye movements. It's more critical during learning. And I guess, and I think that this also kind of actually nicely ties it to the entorhinal cortex and to the model of the grid cells, because this would be more
memory,
sort of memory related or encoding related, and in order to generate future predictions. So if you have all this information kind of properly laid out during learning, then it gives you a good prediction so that you can come up with the identity of the person immediately, right? And you don't need to sort of rescan that face with multiple eye movements. So it's a distinction between more of the encoding memory learning phase versus just sort of someone who's already very familiar.
Great, thanks.
I'd be happy to ask a question if possible.
Mm-hmm.
So first, Galia, thank you for a very interesting and enlightening talk. Thank you. I wanted to ask, and I apologize if it's an ignorant question, but so you assume that impairment in holistic face processing is the outcome of smaller receptive fields, but how do you know that it's not actually, it's not a cause, but how do you know if it's a cause or rather an effect case in terms of so compensatory behavior in CP in trying to recognize individuals, at least from my experience and from what I understand, is to focus on local features or irregular features. And that could actually, in terms of developmental, maybe promote smaller spatial
it's true. Yeah. You're right. So far, we don't know the directionality. I'm suggesting that the directionality is from the small receptive fields that leads to the impairment in holistic perception, because for me, it's like receptive field is sort of the mechanism. It's the physiological property that enables sampling of the environment, and this enables integration. And then holistic perception is kind of the outcome of such normal integration. But you're right. I mean, it's a suggestion. It's a hypothesis. We don't know. One sort of line of evidence that speaks to this and is consistent with the idea that receptive field size is the basis for holistic processing. Comes from, again, studies from Kalani's Spector, and they also looked at children, young children, and they show that receptive field size kind of continue to grow. And again, this is in correlation to, and the size of the receptive field size are in correlation to, are correlated to the behavior. So from the developmental perspective, it's consistent with the notion of this sort of causality that we're suggesting. But in congenital prosopagnosia, we don't really know. It's a working hypothesis. Yeah.
Okay. Oh, interesting. Thank you.
Thanks.
Galia, I also, yeah, as I said, a great talk. I want to ask if you think, I mean, following what the artist said and the idea that Galit asked about holistic processing and face perception, I wanted to ask, since we really usually test in the lab face perception and 2D with 2D information, whereas in real life we are exposed to 3D images, or to 3D stimuli. Sorry, I wanted to ask if you think that the holistic aspect, and maybe also that relates to the grid cells, are at the moment we examine them and measure them in 2D, but they actually represent a 3D structure in space. So do you think that holistic, like the artist said, is actually, normal face perception relies on holistic 3D, not holistic 2D, if you understand what I mean. So integrating this in 3D, which we only tap into with our research in the 2D aspects, but maybe there's 3D, holistic volume-like or object 3D-like representation of faces, which is, that's also lacking.
Yeah, I don't think, I don't know, maybe Galit, maybe you know, but I don't think of any study that used 3D faces in that context in congenital prosopagnosia. It could be sort of another dimension that in which they would show a deficit, but
the 3D will translate to view invariance. You know, if you change view, that you can call it a 3D in some sense.
Yeah.
Yeah, broader.
Yeah, but I think he referred to real life versus images, right? This is how he was trying to explain why he loved painting faces, but it's hard for him in real life. So maybe VR studies, I don't know whether anybody have tried VR.
Yeah, I'm not familiar with such studies, but again, I think what I started with is really that prosopagnosia is something that is evident in real life. So you don't have to, you know, even if you have all the information, like even if you have the 3D information and you have other clues, you still see the deficit. So, but I don't know, you know, what's the kind of the addition, like the delta between 2D and 3D. I don't know of studies that kind of quantified it, and also how it relates to maybe, as Rafi, as you were saying, to viewpoint changes.
Okay, thank you. And I've got another question with respect to the eye movement idea. So I was thinking, I mean, until now, there are no real treatments for developmental prosopagnosia, but I was thinking, and it's not usually, or maybe, I'm not sure which age it's usually diagnosed in, but perhaps this could lead, this concept of yours could lead to conceptual treatments. If you think if you teach prosopagnosics to scan faces in a different way than they actually do, would that improve, or is the hardware limitation going to overcome this, I don't know, the eye movement patterns?
So far, the studies that looked at training in prosopagnosia showed kind of modest success, not great. There's one study with a child that was diagnosed with congenital prosopagnosia, and they claimed that they taught her to change her scanning pattern, and that this actually improved her face processing abilities. but it's like a single child, and in adults the results are very there are some studies showing some improvements, but it's not amazing. So potentially, or I guess maybe once the hardware, as you said, is kind of impaired, it's very hard to come up with alternative mechanisms. So, and you cannot force, for example, holistic processing when there's when holistic processing is impaired, or you cannot make, receptive field larger when they are small. So maybe this has to do with why the success is kind of so moderate so far. But yeah, I mean, it's an important sort of future direction.
Yeah,
and also
inspiring.
Yeah.
Thank you. Micah, do you want to ask something?
Yeah, I just wanted to follow up on that and just say that, well, first of all, thanks again for a super interesting talk. I'm just actually writing you an email as we were discussing. But yeah, I just wanted to follow up on that eye movement thing. I mean, based on the information that I'm aware of, it seems as though our fixation patterns already reflect what our brain can most efficiently discriminate or distinguish. So I would find it very surprising that by training an individual with an impairment, with any given strategy, should lead to any type of improvement, because it seems as though our ocular motor behavior already is trying to maximize information gain with reference to what we bring to the table biologically. I don't know, what are your thoughts to that?
Yeah, I mean, I think I tend to agree, and again, if the mechanism is already impaired, then it's going to be hard to change it, right? Definitely in an adult, and we see in the literature that the success is kind of very minimal. So yeah, I think I tend to agree with your sort of assumption that it's going to be very difficult to see any kind of real sort of progress just by kind of changing the strategy or teaching something else to these individuals. I don't know if that's what you meant, or
yeah, I was just wondering what you thought about that. Yeah, thank you.
Sure.
Okay. If there are no more questions, then again, I'd like to thank you, Galia, for the great talk.
Thank you very much, everybody. Thanks for being here.
Thank you very much. And just want to say, next week, Professor Michal Irani from the Weizmann Institute will be here to talk about deep internal learning, deep visual inference without prior examples. So you are most welcome to join us again. And thanks, Galia. Thank you, everybody, for joining.
Bye, Galia.
Thanks, Sean.
Great talk.
Bye. Thank you very much. Bye. Bye-bye.